everyone, welcome back to my channel. This is lecture 7 of chapter Structure of Chromosomes, Cell Cycle and Cell Division. And in this last lecture, we are going to solve all the diagram based questions of your book chapter Selena class 10. So let's begin. This is the first question. The question says, given below is a systematic diagram of a portion of DNA. So this is a portion of DNA. We can see two strands, okay? Two strands composed of nucleotides. So let's begin the questions, okay? How many strands are shown in the diagram? Okay, we can see this is the one strand and this is the second strand. So two strands are shown. So let's write the answers. For answer A, two strands. Okay, now next question. How many nucleotides have been shown in each strand? It is asking in each strand how many nucleotides. So nucleotide consists of pentose sugar, nitrogenous base and a phosphate. So this is phosphate, this is nitrogenous, this is sugar and this is the nitrogenous base. So this whole comprises one nucleotide. So in each strand we can see 1 and 2 nucleotides, and in the second strand also we can see 1 and 2 nucleotides. So the question says how many nucleotides have been shown in each strand. So each strand has 2 nucleotides. Okay, so for B also 2. Okay, how many nucleotides means 2 nucleotides are shown in each strand. Now next question, name the parts 1, 2, 3, 4 and 5. So let's name the parts. Okay, so 1 is phosphate, 2 is pentose sugar, 5 carbon sugar. So 2 is pentose sugar. Now third is nitrogenous base. We can see third is nitrogenous base. Fourth is what? Fourth is hydrogen bond. It connects the nitrogenous bases of one strand with the nitrogenous base of another strand. So fourth is hydrogen bond. Now fifth is again nitrogenous base. Why we are seeing it nitrogenous base? Because this base has nitrogen present in its structure. That's why we are calling them as nitrogenous bases. So fifth one is again nitrogenous base. Now last question, name the DNA unit constituted by the parts 1, 2 and 3 collectively. It is saying this whole unit 1, 2 and 3 means one is the phosphate, second is the pentose sugar and the third is the nitrogenous base. This whole unit comprises nucleotide. So here the answer for the D part is nucleotide. Okay, I hope this question is clear. Now next, let's move to the next question. The three sketches given below A, B, C, okay, A, B, C are intended to represent the replication of DNA. It is representing the replication of DNA, means the synthesis of new DNA. What should be their correct sequence? It is asking correct sequence, starting with the first and ending with the last. Okay, it is asking the correct sequence, means which step will occur first, then the next step, and after that the next step. So we just have to tell the correct sequence. So let's analyze this given figure. So A shows 2 copies of DNA, B shows a single copy of DNA, okay, and C shows a single copy of DNA which is forming new copy of DNA. Okay, so we can clearly interpret that first there will be a single copy of DNA which will unwind, okay, and form the other copy of DNA. Okay, so suppose this is the single copy of DNA and it has unwinded, and on that on that unwinded strand, it is forming a new copy of DNA. Okay, suppose originally it was like this. The original DNA, it unwinded, and on the unwinded strand, it is forming now two copies of DNA. So after B, C will be occurring, and now it has finally formed the two copies of DNA. So after C, again A will be occurring. So this is the correct sequence: B, then C, then A. Okay, I hope it is clear. Okay, this question says, study the diagram and answer the question. So part A, name the parts 1, 2, 3. We can see here a stage of a cell division. Okay, and from this we have to identify the parts 1, 2 and 3. So let's identify. One is pointing towards centrioles. Okay, so if you are confused whether it is centrioles or centrosomes, just write the point as centrosomes containing centrioles. Okay, this way you will be given the marks on the keyword. So let's write it one as centrosome containing two centrioles. Okay, now part 2. Part 2 is clearly visible as spindle fibers. So part 2 is spindle fibers. Now next, part 3. Part 3 is chromatids. So part 3, chromatid. Okay, so we can see in this given diagram that the sister chromatids had separated and they are moving towards the opposite poles. Okay, so part 3 is chromatid. Now the next question, identify the above stage and give a reason to support your answer. We do not have to just identify, we also have to give the reasons for why it is known as that stage. Okay, so from the diagram we can see that sister chromatids are moving towards the poles. They have split it from the center and we can see furrow in the cell membrane. So this phase actually is late anaphase. So the answer for B is late anaphase. Why late anaphase? Because the sister chromatids have already separated and they had covered a larger distance towards the poles, and we can see a furrow formation. In the initial or the early anaphase, we do not see this furrow. We see this furrow only in case of late anaphase. Okay. Now we have to give a reason to support your answer. We have to give a reason to support this answer, that why it is anaphase. So sister chromatid moving towards opposite poles. Okay, and the second reason is furrow formation in cell membrane. Furrow formation in cell membrane. Okay. Now the last part, mention where in the body this type of cell division occurs. Okay, we have to mention in the body where this type of cell division occurs. So it occurs in the body or somatic cells. Okay, it occurs in all the parts of the body, in all the cells of the body except the reproductive cells. So it occurs in body or somatic cells except the reproductive cells, because in reproductive cells meiosis occurs. Okay, now the last part, name the stage prior to this stage and draw a diagram. We have to name the stage that comes before this anaphase stage and we have to draw the diagram of that stage. So let's do it. I am erasing this part. So part D. So do you remember that mnemonic? Prime Minister at Kolkata. Kolkata was referring to karyokinesis, because these are the stages of karyokinesis, means they are the stages of nuclear division. So before anaphase comes metaphase. So prior to anaphase comes metaphase. So let's write it. Name the stage. So the stage is metaphase, and we have to draw the diagram of metaphase. So let's draw. This is the cell membrane. Okay, in metaphase what happens? The chromosomes arrange themselves in one plane at the equator. So this is the one plane, and here the chromosomes will arrange themselves. So from the diagram we can see four chromosomes. So we will draw 4 chromosomes. Remember this. And here centrosomes containing centrioles. These are the aster rays. Okay. Now let's connect the centromere with spindle fibers. Okay. Now let's do the labeling. So this is spindle fiber. This is aster. This whole is centrosome containing centrioles. This is centromere. These are the chromatids. Okay, I hope it is clear. Now let's move to the next question. The diagram given below represents a certain phenomena which occurs during meiosis. Okay, name and explain the phenomena by using the terms homologous chromosomes, chromatids, and crossing over. So here this diagram shows a phenomena which is taking place during meiosis, and we also have to explain this by using the terms like homologous chromosomes, chromatids, and crossing over. So we can see in this that their genetic material is being exchanged. Okay, here. So this process is known as crossing over. So the phenomena here is crossing over. This is the phenomena. Now we have to explain. So what happens in crossing over? One set of chromosomes comes from the mother and the other set of chromosomes comes from the father. So which comes from the mother is known as the maternal chromosome, and that comes from the father is known as the paternal chromosome. They come together, okay, and they form homologous chromosomes. What are homologous chromosomes? Chromosomes which are exactly of similar shape and size. So they form homologous chromosomes, and between them the genetic material get exchanged, means they contain a set of genes which may be for your hair color, for your height, for your eye color, so that they get exchanged between the two chromatids. Okay, and these chromatids are non-sister chromatids because one chromatid is of the mother maternal chromosome and the other chromatid is of the paternal chromosome. So in them the genetic material is getting exchanged. Okay, we can see here the red part has come to this one and the black part has come to this one. Okay, and here a X-shaped structure which is also known as chiasma is formed. So let's explain crossing over. So the exchange of genetic material between the non-sister chromatid chromatids of a homologous chromosome is known as crossing over. Okay, so we have used the terms like homologous chromosomes, chromatids and crossing over. So this explains this whole phenomena. If you want, you may also write about chiasma. So chiasma is a egg-shaped structure which is the point of attachment between the two non-sister chromatids formed during meiosis. Okay, so you may write about chiasma also. I hope this is clear. Okay, now let's move to the next question. Question says, examine the diagram and answer the question. So we have to observe this diagram and answer the following question. So the first part says, identify the stage. Okay, give one reason in support of your answer. So we have to identify this given stage of cell division. So in the diagram we can see that nuclear membrane is disappearing, first point. Then we can see the centrosomes have reached the opposite poles. Okay, and we can also see that chromosomes are clearly visible, they have become distinct. Okay, and the spindle fibers have formed between these two centrosomes. Okay, so this stage is late prophase stage. So let's write the answer. It is late prophase stage. Okay, now we have to give one reason. So we may write this, chromosomes have become distinct. So chromosomes have become distinct. If we want, we can write one more reason, that nuclear membrane is disappearing. So the second reason is nuclear membrane. We can see this. Okay, nuclear membrane is disappearing. Okay, now let's move to the second part. Name the cell organelle that forms the asters. Okay, this is the rays that are coming from the centrosome is known as aster. So the question is asking the organelle which form this aster. So centrosomes containing centrioles, they form the aster. So let's write centrosome containing centrioles. Okay, they form the asters. Now next part, name the parts labeled 1, 2 and 3. We have to label the parts 1, 2 and 3. So I am erasing this answer. So part C. First is, okay, we can see that first is pointing towards the centromere. The point of attachment of the two sister chromatid is called as centromere. So first is pointing towards centromere. Okay, now second is pointing towards the chromatid. Second is chromatid. Okay, now third is, we can see third is spindle fibers. Okay, this third is spindle fibers. Which have formed between the two centrosomes. Okay, now next part, name the stage that follows it. Okay, it is asking the stage that comes after this stage. So which stage comes after prophase? That is metaphase. Okay, now how is that stage identified? And we have to give a reason that how we identify that stage. What is the feature of that stage, that this stage basically refers to metaphase. So again I am erasing this answer. So for D part, The stage that follows it is metaphase. Okay, now how is that stage identified? So in metaphase, what happens? The chromosomes arrange themselves in one plane at the equator. So let's write the reason. Chromosomes arrange themselves in one plane at the equator. Okay, so this is the reason. Now let's move to the next part. I have taken the E part separately. So let's see. Mention two differences between mitosis and meiosis with regard to. We have to mention two differences. Okay, with regard to mitosis and meiosis. The first point is the number of daughter cells produced, and the second point is the chromosome number in the daughter cells. So let's do it. Here we are writing mitosis and meiosis. So the first point says with respect to the number of daughter cells produced. So in case of mitosis, we know that 2 daughter cells are produced, and in case of meiosis, 4 daughter cells are produced. So let's write here 2 daughter cells. And meiosis, 4 daughter cells. Now next part, the chromosome number in daughter cells. Okay, so in mitosis we know that the daughter cells have the same number of chromosomes as that of parent cell. Okay, so if parent cell is diploid, means it has 2n number of chromosomes, the daughter cells will also have 2n number of chromosomes. And in case of meiosis, the chromosome number is halved with respect to the parent cell. So if the parent cell has 2n number of chromosomes, the daughter cells will have n number of chromosomes. So the chromosome number in daughter cells. So daughter cells will have, here daughter cells will have 2n diploid number of chromosomes. Okay, and here daughter cells will have n number. N means haploid number of chromosomes. Okay, we have covered all the parts of this question. So now let's move to the next question. The question says, given are 3 diagrammatic sketches A, B, C. Okay, we have to see this 3 diagram sketches and we have to answer the following questions. Okay, so first is identify the phase. So this 3 phases, it represents one stage of a cell cycle. Okay, with 3 different diagrams. So let's see what is happening. We can see clearly in all the 3 diagrams that the chromosomes have aligned themselves at the equator in one plane. So this happens only in case of metaphase. So the phase is metaphase. Now next part, what is the diploid number of chromosomes shown? So we can see in each diagram there are 4 chromosomes that are shown. So the diploid number of chromosomes that are shown is 4. Okay. Now next question, identify animal or plant cell. We have to identify whether A, B or C are plant or animal cell. So A we can see only cell membrane, we can see centrosomes, that's why A is an animal cell. So A is animal cell. In case of B also we can see centrosome, cell membrane. So again B is also animal cell. Now in case of C we can see outer boundary which is known as cell wall, then cell membrane, and inside we can see that centrosomes are absent. So it happens only in case of plants. So C is plant cell. Now next question, which of these is are shown in correct direction? Okay, we have to identify whether A, B and C they have the correct orientation of spindle fibers or not, and whether they represent correctly metaphase or not. So options are only A, only B, only A and C, all of the three. Here if we observe the diagrams one by one, we will see that in all the diagrams everything is being in correct direction. See, here the chromosomes are arranged and the spindle fibers are joining the centromere and they are originating from the centrosome. So A is also correct. In case of B also, it is also correct. We can see this spindle fibers joining the centromere of the chromosome, and the chromosome are arranged at the equator plane. So B is also correct. This C is also correct. Both B and C, they are given in your book. Just go and check the metaphasic stage. Okay, this C part has been rotated. Okay, in your book it is like this, but here in the question it is rotated like this. It is the same diagram which is given in your book. So both B and C are taken from your book only. Okay, so here also C is also correct. Only in this case the cell has rotated 90 degrees. So here also we can see the chromosomes are attached with the spindle fibers through the centromere. So both A, B and C are correct. So option 4 is correct. All of the three are correct. Okay, now let's move to our last question. Below are 4 stages A, B, C, D. So these are the 4 stages of a cell division. A, B, C, D. Okay, and we have to answer these questions. So first part says, is it a plant or an animal cell? Give 2 reasons. Okay, so we can clearly see that only cell membrane is present plus centrosomes are present. These centrosomes are absent in plant cell, and cell wall also have cell wall as the outer boundary. So here only cell membrane and centrosomes are present, so it is an animal cell. Reason, only cell membrane present, or you can write cell wall absent. Okay, you can write cell wall absent. Now next reason, centrosomes present. Centrosomes present. Okay, now question asking, is it undergoing mitosis or meiosis? So we can see that here mitosis is going because we are not able to see homologous chromosome. In homologous chromosome, 2 set of chromosomes must be aligned, must be present together. So here only we can see 1 set of chromosomes. Okay, so it is going mitosis, and we are not able to see crossing over also. That crossing overs occur in case of meiosis. Okay, now what should be the correct sequence? So from A, B, C, D we have to arrange them in proper sequence. Okay, so in A we can see that the sister chromatids have separated and they are moving towards the opposite pole. So it is representing anaphase. Okay, and in B we can see that the chromosomes are trying to become distinct. Okay, and we can see 2 centrosomes that are trying to move towards the opposite poles, and it occurs in case of prophase. Okay, in C we can see that these chromosomes, they are trying to arrange themselves at the equator plane. So it is representing transition to metaphase. Means after prophase these chromosomes are trying to arrange themselves at metaphase. So it is representing transition to metaphase. And in D we can see that The chromosomes are properly arranged in one plane at the equator. So D is representing metaphase. So we have to arrange them in proper sequence. So our sequence will be B first because it is representing prophase. Then C because from prophase it is going to metaphase. So it is representing transition to metaphase. So B, C, then happens our metaphase, and after metaphase the sister chromatids splits from the center and they move towards the opposite poles in case of anaphase. So let's write, first comes B, then C, then D, and last comes A. So this is the proper correct sequence. Okay, now name the stage that precedes the earliest of these stages. It is asking before all these four stages, which stage comes the earliest. So that stage is known as interphase. Okay. Now, last part says draw the stage named above inside the blank space provided. So in the last part, we have to just draw the stage that we named in the previous part question. So draw the stage, okay? So the stage is interphase and we have to draw that in this blank space which is provided. So let's draw the interphase stage. So this is the cell, okay? In interphase stage, this is the nucleus, okay? We see chromatin fibers, okay? We see chromatin fibers and we see nucleolus, and here we see the centrosome. Okay, so let's label it. These are chromatin fibers. This is nucleolus. This is nuclear membrane. This is cell membrane. This empty space is cytoplasm. This whole is centrosome. Centrosome containing two centrioles, you may write. Okay, so this is the last question of today. Before ending the lecture, I really want to remind you of one thing. I want to remind you to stay strong on difficult days. Okay, there will be days when you will feel low and you will not feel like doing anything. So just keep on reminding yourself that you will get out of this, you will get out of this. So on those days, stay strong. Don't feel guilty. Just hold your hand, being present with yourself. Okay, and take your time and keep moving forward. So that's all for today. Take care. Thank you so much.